Greetings. My name is Katie Kimberling, proud member of Team 8 in USD MADS 599. Today, we introduce our proposed Capstone project entitled Breaking the Lead Optimization Bottleneck in Drug Development for Emerging Infectious Diseases. My teammate, Jordan Torres, brings a phenomenal background in computer science to the table. Lyndon Conrad Marut offers chemical engineering expertise and work experience in the pharmaceutical industry, and I provide insight and interest into infectious diseases and epidemiology from a public health perspective. We have an arms race going on, and humans erroneously believe we are bigger and smarter, but we are being historically dominated by organisms so tiny they cannot be seen by the naked eye. An emerging infectious disease, or EID, is one that either has appeared and affected a population for the first time or has existed previously but is rapidly spreading, either in terms of the number of people being infected or to new geographical areas. Typically, these are zoonotic diseases, almost always viruses, that have existed in an animal or insect reservoir and jumped into or spilled over into a human host. Bubonic plague in the Dark Ages is a very well known example, but more recent events include Ebola in Central Africa and Germany, Zika virus in South America, hantavirus in the four corners of the southwestern United States, and the most recent COVID-19 global pandemic. As population growth increases and humans encroach into new territories, they are progressively exposed to animal populations, and the likelihood of viral zoonotic spillover is simply a matter of time. To combat the inevitability of an EID, we need broad spectrum antivirals, or BSAs, at the ready. But discovering and creating drugs is neither quick nor cheap and can take up to a decade and millions or even billions of dollars to go from a hit to a lead. A hit is a molecule that proves effective against a target in a lab, but this might be too large to fit into pill form for a human to consume, or too toxic, or not physiologically soluble. A lead is a molecule that is safe for human consumption that effectively targets the intended virus. We live in a remarkable time. Data science can potentially use the tool of machine learning to harness the computing power of generative AI to exponentially shorten the time span from hit to lead, saving countless lives, billions of dollars, and years spent in trial and error, hit or miss in the lab. Recent history has demonstrated low enthusiasm and scarcity of growth in the field of infectious diseases. To wit, for bacterial infections, every new antibiotic brought to market over the past few decades has been only a slight variation on existing drugs discovered before 1984. While our project will focus on BSAs, antivirals, only one of the top 50 pharma companies has antibiotics in clinical development, and nearly 75% of the companies currently developing antimicrobials can be regarded as pre-revenue with no approved products in the market. Market analysis has shown that drug resistant forms of these diseases will grow significantly by 2025, with very few new drug strategies in the pipeline. Because many emerging infectious diseases share similar structural biological targets, like polymerases or cell surface receptors, we hypothesize that generative AI can be exposed to current broad spectrum antivirals, trained on them, and prompted to propose new novel structures that will shorten the time from hit to lead.
The primary objective of our Capstone project is to design novel antiviral molecules using GenAI. So, traditional lead optimization can take years, so we're leveraging AI to try and explore the chemical space more efficiently and identify structurally valid novel candidates. So, to do this, we'd like to train a GenAI model on approximately 50,000 SMILE strings of broad spectrum antivirals collected from Chemable and PubChem. Smile strings are just the chemical structure in string form. By learning structural patterns associated with drug-like behavior and biological activity specifically, we hope the model can generate optimized analogs of known seed compounds. From a technical standpoint, our pipeline would begin with data preparation, where we would clean and standardize the SMILE strings, then train a Gen AI model on them, which will most likely be a variational autoencoder or a transformer to learn those latent chemical representations and generate new molecular structures. We would then evaluate those generated molecules and validate the outputs using metrics like structural validity, novelty, and similarity, and integrate the entire pipeline into an interactive web app. I'll talk about the pipeline in more depth next. In terms of our technical stack that we've discussed, we'd like to use RDKit for molecular processing, PyTorch or TensorFlow for model training, scikit-learn for property prediction, and Streamlit for deployment. To do this, our team has proposed a data pipeline, which begins with cleaning and tokenizing the SMILE strings using RDKit to ensure the molecular representations are standardized and machine readable. Next, we would train the Gen AI model, which again would most likely be a variational autoencoder or a transformer, to learn the structural patterns from known antiviral compounds. To evaluate the quality of the Gen AI model, we'd use three validation metrics. First, percent chemically valid molecules. This measures how many generated SMILE strings correspond to real and physically possible molecules according to chemical rules. Second, percent novel versus the training set. This would tell us how many generated molecules are truly new and not simply duplicates of compounds the model has already seen. Third, Tanimoto similarity to the seed molecule. This quantifies how structurally similar a generated molecule is to the original seed compound, allowing us to balance the innovation with actual biological relevance. After training and validation, we would generate the new molecular analogs and run additional chemical structure validation using RDKit to confirm the proper valency, ring closure, and syntax. This is where my chemical engineering background would come in. We would then evaluate drug likeness using predictive models. Toxicity is treated as a classification problem. Solubility would be treated as a regression task, and drug likeness would be assessed using Lipinski's rule of five. Finally, we would rank all candidate molecules using a composite drugability score, which integrates these predictive outputs into a single prioritization metric. Overall, this pipeline ensures that generated molecules are not only novel, but chemically valid, biologically plausible, and computationally optimized before moving towards experimental consideration.
What we expect from this project is not just theoretical model output, but actionable molecular candidates that directly address the lead optimization bottleneck in zoonotic infectious disease drug development. Our generative pipeline is trained on known broad spectrum antivirals represented as SMILE strings. By learning structural patterns from validated compounds, the model can generate optimized analogs, predicted to be more soluble, less toxic, and chemically valid. The key impact is speed. Instead of taking years to refine a promising hit into a lead candidate, our system can generate hundreds or even thousands of computationally scored molecular upgrades. This tool does not replace laboratory science. Researchers will still need their own hypothesis and candidate molecules. Our system acts as an enrichment layer, evaluating proposed structures and generating optimized alternatives to improve properties like solubility and toxicity. The goal is not to directly discover, but to accelerate refinement and help move from hit to lead more efficiently. Ultimately, this project shifts drug discovery from reactive to prepared. Our project timeline is structured to reflect a realistic end to end data science workflow, moving from data acquisition to deployment. In weeks 1 and 2, we focus on data acquisition and pre processing. We collect antiviral compound data from Chembl and PubChem. We then retrieve SMILE representation. Finally, we clean and validate them using RDKit to ensure chemical legality and structural consistency. In weeks 3 and 4, we build the data pipeline and train our generative AI model. This involves teaching a SMILE-based transformer, or VAE, the grammar of chemical structures so it can meaningfully learn molecular patterns from known broad-spectrum antivirals. Week 5 is dedicated to inference and scoring. We generate new molecular candidates and pass them through computational filters such as Lipinski's rule of five to evaluate drug likeness and prioritize viable analogs. In week 6, we construct and refine the web artifact, integrating visualization, scoring outputs, and user interaction. Finally, in week 7, we consolidate our findings, finalize the paper, and refine the presentation.